Unlock the future of pathology with Pathology News, your ultimate source for cutting-edge insights, expert analysis, and the latest breakthroughs in digital and computational pathology. Don't just stay informed, stay ahead.
Yeah, I wanted to move the conversation on to scanning. Now, we've obviously talked about the different modalities that you've got here, but yeah, from my understanding, you've got kind of 3 distinct scanning platforms at the core here at the Cambridge Institute. So I was hoping you could kind of walk us through those, their capabilities, and yeah, what each scanner is geared to do.
So actually, we've got 4 now.
Okay.
So we've just acquired a new baby.
Yeah.
Mainly because so we've been using the Aperio 82 for bright field scanning for over 10 years now, but it's getting to the end of its life. So if it breaks down, we won't be having to replace any parts.
Okay.
We won't be able to replace any parts, and so we need an alternative.
Yeah.
And that's when we started scoping what was in the market and having conversations with vendors. So. Going to conferences like Digital Pathology conference was quite useful because we've got an array of different vendors and equipment there that we could just,
Yeah, we turned up with slides and we were like,
yes,
okay.
and test
them. We literally turned up with a box of slides and we're like, can you test these out?
Okay.
Like a preliminary test to
absolutely.
then see which ones we would take forward to demo. So on the back of that, we just acquired an Olympus VS200 to potentially replace Aperio.
Yeah. I'm interested by the, so you turned up to a conference with a box of slides. And what was important to you from the scanning perspective?
So basically, because we work in a research setting, it's not diagnostics, we need some flexibility. So we prime like image quality, color accuracy. That's because we obviously need to make sure that the downstream analysis is reliable. So we just need to make sure that we can scan the tricky samples we have. So we have an array of different sample types, things that are difficult to find on the slides, like isolated cells, fatty tissues, TMAs, things like that. So we just needed to make sure that we could cover all of those samples, have the flexibility to tweak settings and adjust focusing, because that's one thing that we have found that most scanners that are being built for diagnostics to be quick don't allow so much that flexibility.
So off the back of that, did you end up with a short list of scanners that you then wanted to demo?
Yes, so we started by coming up with a list of criteria we wanted to meet. So things that we definitely needed to, boxes that we needed to tick. Yeah. And then going from there, we kind of excluded the ones we wouldn't think would do the job, basically. So we shortlisted that to 4 scanners to demo. And then we just contacted the companies and got them to bring them the scanners in, so we could fully test them to its limits, pretty much, to see if you would integrate in our workflows.
Yeah, I mean, this is probably something we don't talk about enough on the channel, but that seems like a really roundabout way to do something like that. You know, how do you procure a scanner? You know, bringing them all into your lab and having them demoed. Great idea.
The images that are always provided on brochures and things like that are always like optimally stained, absolutely beautiful with like, you know, everything. That's not research. That's not what we have to deal with. Yeah. So we need to physically
test them in
their
own
test the machines. Yeah.
And obviously we want some speed. We want throughput. So we want slides to be scanned, you know, quickly enough. But ultimately, you need good quality. So that's what we were looking primarily. And things like mega scanning capability, Z-stacks, so all of those things that needed to tick those boxes. So
your 3 main tick boxes for scanning then are throughput, quality, and speed, would you say?
Not so much the speed, because what happened was that we happened to choose one that wasn't the quickest one.
Okay, yeah.
So I would say also compatibility with downstream image analysis software was a big one. So we needed to make sure that the file formats were supported on those softwares. So I would say that image quality, flexibility, definitely being able to manually scan slides and tweak profiles.
If you've ever scanned like just in situ hybridization, just duplex in situ, so you've got your hematoxin and your 2 probes, and your probes are on a 3 micron thick section, your probes are still never on the same Z plane. So having an ability to manually focus or have Z stacks was sort of almost a non-negotiable, which then sort of tracks away from what, say, a more clinical high throughput lab would want, because that would slow everything down, and that's just not what they need.
Yeah, and because we got this scanner as well, so it came with a fluorescent capability. So I didn't mention our fluorescent scanners. We have the AxioScan, Zeiss AxioScan Z1, that is capable of scanning up to 5 colors. Then we later acquired the PhenoImager HT from Acquia, now Quantrix, that can scan up to nine colors and spectrally mix the images. And obviously, we always struggle a little bit with this question about detecting those ish spots. So you wouldn't be able to do any Z planes to capture the full range. And it was always a big frustration for us. So we are hoping that, obviously, this scanner wasn't acquired for the fluorescence, it was for the bright field, the VS200. But given that we have that bonus, we hope that this can open some other doors and help with scanning more complex things.
Yeah, it's going to make our downstream analysis for in situ hybridization easier.
So much
Cara.
easier.
And would you say that's the scanner that you use most now, or is it a mix and match?
So I would say that what we have the most is bright field slides.
Yeah.
So currently that's the Apereo 82 that we are using. Hopefully we'll be moving on to the VS200, and between the 2 fluorescent scanners, the PhenoImager HT. So workloads are increasing dramatically for fluorescence.
Multiplex, so 6-plex upwards,
Yeah.
which makes the AxioScan not.
I mean,
not
I love the AxioScan, you know that. The resolution is great.
Image quality, beautiful.
Flexibility, it's like a microscope, a really good microscope in a box. But yeah, it has its limitations in terms of how many markers you can image. And it doesn't spectrally mix the images, so yeah. So basically, depending on the project we have and what you want to get out of it, you would choose the scanner.
Yeah, and I imagine they look really different, and I think that's something we're going to come on to in just a moment. As color standardization, slides looking very different once they're scanned. So it sounds like from the procurement perspective, you guys went about it in a really good way. I was wondering if you had any advice for other labs looking to procure a scanner.
I would say set your priority list very well. It's quite important. Speak to vendors, demo the instruments if you can in-house. If not, there is the NPIC in Leeds, which is the National Pathology Imaging Cooperative,
Yep.
I think. They have an array of scanners, so they could help potentially and give you some advice, scan some slides for you. And I think it's really good to start looking into things well in advance and make a good record of the findings you have. Because for us, we had to fill out forms, have meetings, and it was like a really long process. And you need to justify really well why you want to purchase this particular one. So basically, I mean, it does go through, the process goes in a way that you kind of present the findings and then it gives you an answer of which one you want to choose. It's not that you go and say, I want this one. But obviously just have it all very well established in terms of the criteria you need to meet. I would say. Not sure if
Talk to people.
talk to people. Yeah.
Contact, speak to the vendors and say, oh, are there any other labs in the UK that have got this scanner or this bit of equipment? And then ask, would it be okay to be put in contact with them? And speaking to other people using equipment, you get a lot more information than you would from the vendors.
Yeah, absolutely. And there's a lot of information out there now, isn't there?
Yeah.
I know the ESTIT team in Europe, they do loads on this kind of thing. I think they have scanner comparison, probably quite similar to what you've done actually, but it maybe doesn't look specifically into individual labs. And what they need is more of a general ability of each scanner. Now, I wanted to move the conversation on and talk about colour standardisation, which I promised we would do. So, obviously this is quite a new field. We're only adopting these technologies at scale now, and we're running into a few hurdles that perhaps we didn't expect to at the start. And one of those issues is colour standardisation. So yeah, to start with, and for anyone that isn't familiar with this issue in scanning, could you tell us a little bit more about that?
So there can be a lot of variation in the RGB values between the different scanners. And this is something that we currently see with the 4 different scanners we have, because the 4, even the fluorescent scanners we have the bright field capability. Cara will be able to talk a bit more about how much this impacts the downstream analysis. But basically, calibration, we think, is key, and I think it's really good to have good standardization of the images and try to narrow down these differences as much as possible for downstream analysis.
Yeah. Every scanner, so all scanners have different objectives, they have different cameras, they have different light sources, all of which give you variations to have to sort of contend with. So the same slide scanned on for all 4 of our different scanners
will
looks totally different, like the colour is very different. But then the same slide scanned on the same scanner within 2 different institutes, for example, could still look different, just down to like maintenance and things like that and how it's looked after. So, and then you always get tissue variability as well. So, how well it was fixed, the protocol that was used, for example, like the H&E, so many variations of how you do the basic chemotoxin and niacin stain, and having to contend with that is fun.
Yeah. And you mentioned degradation of light sources there. So on the same scanner scanning perhaps the same slide over a different time frame, let's say you did it once in 2025 and once in 2026, would you see a visual difference in RGB? Yeah.
I would say that's probably actually true. So we've had our 82 for 10 years, and actually I think if you looked back at sort of images that were acquired 10 years ago, or probably not quite 10 years ago, but sort of when the light sources were new, and then we've probably had a replacement light source since then. It's probably a whole new machine inside it actually. But yeah, we'd probably be able to see the difference. It's quite noticeable in things like special stains, so mass on trichrome stains that we get. These are manually stained. We have control samples that go in with every single batch of slides that we stain. They all look very similar, but obviously you do get that variability, and actually to be able to find that blue against the purple that's in that stain is quite difficult, and to quantify that is painful sometimes.
It's been problematic, let's put it that way.
So if we used AI, for example, we can sort of account for that variability. So as long as we give examples to our protocols or to the software, if we give it examples across multiple different sort of staining batches and things like that, train it to understand that this shade of blue is the same as this shade of blue, which is the same as this shade of blue, and this resolution and this resolution and this resolution.
Sure.
You can build up a robust enough algorithm to then find what you need to find. But without having that AI capability and that trainability, and also having the examples to give it and time, you wouldn't be able to have that. But I would say that AI is a potential way of helping get across that sort of variation you get.
Yeah, so is this something you're looking to do internally?
Yes.
Okay.
So it's something that I'm currently working on now, in that again, it's matte on trichrome. It's one of my problem stains. But just looking at the variation in blue, so looking at different sort of image analysis platforms. So testing all the both Halo and the Visiofilm software that we have, but also looking at QPath with it as well, and to see if there's a pipeline that we can create to sort of help get rid of that.
Sure.
But then also there are other options that you can use. There's standardisation sort of slides that we can potentially get. So PathQA.
So if you do standardize the actual image to start with, like by calibrating the scanners to a standard, I think that's the aim. So we, as Cara said, we are looking into this potential collaboration with PathQA, or finding a way of potentially calibrating this the 4 scanners we have, and see whether the result will allow us to maybe use each one of those 4 scanners, even for bright field scanning if we need to. If it comes to obviously the file type will be different between them, the resolution will be different, but ultimately, like if you could just reduce as much as possible that variability across them all, this will improve things and make Cara's life easier as well.
But also, as we're moving from the 82 to the VS200, it will help sort of
make it comparable,
smooth that process over. Because if you started your project on one machine, you would like to finish it in theory, but if we have no control on how long the 82 is going to live for, so we want to move everything across as smoothly and happily as possible.
With all of that in mind, what are the downstream impacts of colour variation?
Yeah, it just makes sort of the analysis aspect a lot more difficult. So you end up with, you're either, which is not ideal, you're creating multiple algorithms to analyse different slides of the set for the same answer, which, okay, blue is either blue and red and things like that. It's either there or it's not. But also that gives you the flexibility to sort of maybe be slightly biased as well. So you do have to be quite careful and standard. And I guess when we're doing it as a service, we don't always know that much detail about your samples. So we kind of we will find blue if that's what we're looking for example. And we are less biased than if the researcher was doing it, for example, because they know what they want from the outcome, whereas we don't know what we don't, not that we don't care, but we don't know.
We're unbiased.
We're unbiased,
Yeah, you're just providing the results.
Yeah, so we're providing results however they come. But I do think without AI, that colour standardisation has caused problems. And also things like hematoxin and eosin, pink and purple, their RGB values are so similar that actually to distinguish between the two is difficult. But, it is difficult, and everything has an H&E. So, there's so many H&E sort of slides and images, and there could be so many sort of things that could be done with those images. But obviously, pink and purple are difficult to work with. So, it's just introducing that AI will help vastly, I believe.
Let's talk a little bit about the future then. Obviously, thanks to AI, we're developing all these other new biomarkers that AI can see, but the human eye can't. I'm interested, do you think we'll see better integration between all of these different imaging modalities with all the data coming together in order to support not just diagnostics, but research as well?
Yeah.
What are your thoughts on that?
Yeah, so sort of collecting all of those images, all of the sort of the data that goes with them, any results that came with them, any information that they have about the samples, things like that, all of that being collected into one place, and then using that data to sort of mine for further details, training different sort of AI programs to then be able to find patterns that are there that we would never see, or find sort of a marker that would be of interest that would just be missed. That's what sort of that's the sort of direction the Institute is going in, sort of having that sort of collaborative, having everything together in one place.
We don't know how the technology is going to move on, and in a year's time or 5 years' time, what are we going to be able to do with that data? What questions might we be able to go back and ask? Like you said, mining that data in the future. The things we don't even know yet.
Yeah. So, is that something that the Institute is focused on? How are you bringing all of that data together?
there's certainly an initiative to sort of bring that all together. What it looks like yet, you know, it's evolving, but yeah, certainly it's kind of very large on the agenda.
So thinking about the future then, you know, what technologies do you think will have the biggest impacts on what you guys do?
Like Elsa said earlier, we know we're always scanning the horizon, looking for those new technologies and trying to kind of, you know, to sort of preempt what the needs of our researchers are going to be, and to sort of test those out, you know, ahead of time so that we're ready for when those technologies are here, we can adopt them. But it's difficult to say, isn't it? You know, we are moving kind of in quite a high-plex, you know,
environment. I mean, if you think about how quickly that high-plex has changed from when you used to think high-plex would be sort of 5 colours, it's now 5 is tiny-plex. It's, you know, it's moved so quickly. So to see where it's going now, I personally can't see it going more higher plex. I feel like where else I mean, maybe we could, I don't know, but it feels like you're only scratching the surface of the data we're getting out of this high-plex multimodal sort of the analysis that we're doing. Like, there's probably so much information in there that we're missing that actually...
I feel like there's a lot of analysis of that data yet to be done. And I think there is going to be like maybe a push back to a bit lower plex, maybe, because I think there's going to be some validation required of some of those, you know, those hits, for example. They're going to perhaps come, you know, maybe as that data is explored, there's going to be a bit more low plex kind of work going on alongside it.
I think that's a good point, and I think the fact that we don't have any standardization for fluorescence and this, you know, the same thing that we were talking about color accuracy for bright field. I think this is something important that needs to be addressed as well. And I think building massive amounts of data that might not be completely accurate or you might not be able to interpret later could be dangerous as well. So I think, as you said, maybe lower plexity, but integrated with AI tools, innovative AI tools that would make more predictive models, maybe. I think that's how things are going, potentially.
Thank you. And, you know, just to finally wrap this up then, kind of a last question for all of you. You know, do you have any advice for any very young researchers out there who are just getting into this field or perhaps, you know, working on their own research projects?
Yeah, do your controls.
Okay.
Always have a control sample, have a positive and a negative control. That will then, one, prove that your experiments worked, but also then can be used in your analysis pipeline to be like, okay, so this is what negative looks like, this is what positive looks like, and then you can sort of scale everything in between. You've got a yes and a no, and then the results and then in between.
That is definitely something that, you know, over kind of built up over years of experience. Conduct a pilot project, you know, a pilot study. You know, check your end-to-end pipeline. Does it work? Do you get out what you need to? Can you, you know, yes, can you stain it? Have you validated your antibodies, your probes, your, you know, tissue pre-treatments and so on? Have you done all of that, and can you actually analyse it at the end? Like, you know, can you scan it? Yes. You know, everything. It's really important before you just kind of look at the stain and go, oh yeah, I think that's fine, and then you actually try and analyse it at the end. Just check it on a small select group of samples. With like Cara says, with controls, you know, your positive and your negative, and
we realise it's expensive, and I get it's expensive, but without the control, you need proper validation.
Yeah.
You need a proper question to be answered, so you need to know exactly what you're looking for to then be able to build this pilot studies and you know, just get something out of it. Because if you're just trying something without knowing exactly what you're doing and then trying to take any conclusions out of it, you might just be wasting time, precious time and resources.
Just because you can see it on a single plex doesn't mean when you multiplex it, it's still going to look the same. You're going to have so many different things sort of affecting that marker, for example. So
What I'd also say is, go out there and look for some people who you might think of as experts who can help you. And
People love talking about their
failure is
own
also
work.
a learning opportunity.
Conversations,
Yeah.
questions.
Yeah,
Thank
that's
you.
right.
I suppose the digital pathology, spatial biology community is quite niche really, and everyone likes talking to each other about what it is that they do and trying to help each other as well. So perhaps that's some additional advice on top of yours, Julia. Yes, thank you all very much for that. It's been a real pleasure having you on Pathology News today, and hopefully we'll have you on again in the future.
Thanks very much.
you.
Thank you.
Thank you.
Bye.